Hello friends, welcome to PVIC Breakdowns. In this video, I'll be discussing the structure of DNA. Firstly, what is DNA? DNA stands for deoxyribonucleic acid. DNA in simple terms is the hereditary material found in the cells of living organisms. And what I mean by hereditary is that it is passed on from parents to offspring. DNA acts as the repository for all genetic information. This means that, like an instruction manual, DNA contains all the information necessary for the development and functioning of an organism. It is the DNA that determines the gender of an offspring, the color of our eyes, our genotype, our blood group, and so many other characteristics. So now we are done with explaining DNA in simple terms. Let's give a more biochemical definition of what DNA is. As a biochemist, DNA is basically a polymer that consists of many repeating units or monomers called nucleotides. Now, the structure of the DNA was discovered by James D. Watson and Francis Crick, and the work of these scientists led to our present-day understanding of the DNA. After all, school no be scam. Now, the Watson-Crick model for the structure of the DNA is the widely accepted and currently used model. According to this model, the physical structure of the DNA molecule is basically a double-stranded helix, or you can simply say a double helix. So you have a helical strand and another helical strand, which gives you a double helix. Now, we mentioned earlier that DNA consists of many repeating units called nucleotides. What are these nucleotides? Nucleotides are the building blocks of DNA, just like monosaccharides are the building blocks of carbohydrates and amino acids are the building blocks of proteins. Now, a nucleotide consists of three major components, one being a 5-carbon sugar, which in the case of DNA is deoxyribose. Deoxyribose. You can see 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. This is a 5-carbon sugar. 2 is a phosphate group. We can also call this phosphate group 5 prime phosphate group, since it is attached to the 5 prime carbon of this pentose sugar. And 3 is a nitrogenous base. Which can be adenine, guanine, thymine, or cytosine. Now, before we move further, I want to make a clear difference between a nucleotide and a nucleoside. Like we said, a nucleotide consists of three major components, which include a pentose sugar, a phosphate group, and a nitrogenous base. Now, if we remove this phosphate group, what we now have is a nucleoside. So in order to easily remember the difference between a nucleotide and a nucleoside, we can summarize it in this way. For a nucleotide, we have sugar, base, and phosphates. While for a nucleoside, we have sugar and base only. So these are the three components of a nucleotide, and these are the two components of a nucleoside. Now, the 5-carbon sugar in DNA nucleotides is called deoxyribose because it doesn't have a hydroxyl group attached at the 2 prime carbon. Instead, it just has a hydrogen atom there. So A ribose sugar, which is found in RNA, consists of a hydroxyl group attached at the 2' carbon, while a deoxyribose sugar, which is found in DNA, does not have a hydroxyl group attached at the 2' carbon. So you can see how DNA got its name, deoxyribonucleic acid, from deoxyribose. Now, we have two kinds of nitrogenous bases. We have the purines and the pyrimidines. Purines consist of two ring structures that are fused together, while pyrimidines consist of only one ring structure. And adenine and guanine fall under the purines class, while thymine and cytosine fall under the pyrimidines class. So from school chemistry, we should know that this point in this nitrogenous bases represents carbon atoms which are not shown here. And we use normal counting numbers to number the atoms in this nitrogenous bases. So here we have 1, 2, 3, 4, 5, 6, 7, 8, and 9. And in order to make a difference, we add prime. This is the symbol for prime to the numbers when numbering the pentose sugar. So here we have 1', 2', 3', 4', and 5'. Now, this is the physical structure of the DNA molecule, and this is a detailed representation of this structure. You can see the complementary base pairs just as it is here, and you can see this helical strand, and this helical strand are represented by this and this. And this is even a more detailed representation of the DNA molecule. Now we say that the nucleotides in the DNA strand are linked to each other through phosphodiester bonds, in which the 5' phosphate group of one nucleotide unit is attached to the 3' hydroxyl group of the next nucleotide. So here we have, this is a nucleotide unit now, and we have the 5' phosphate group of this nucleotide joined to the 3' hydroxyl group of this nucleotide through this phosphodiester bond. Now we say that the two DNA strands are antiparallel to each other. That is, they run in opposite directions, with one strand running in the 5' to 3' direction and the other strand running in the 3' to 5' direction. This means that the starting here has a free 5' phosphate group and ends in a 3' hydroxyl group, which in this case is attached to successive nucleotides until we get to this point where we now have a free 3' hydroxyl group. So we can see that this DNA strand runs in the 5' to 3' direction. Now in this strand, the starting has a free 3' hydroxyl group which ends in a 5' phosphate group, and in this case it is attached to successive nucleotides until we get to this point where we have a free 5' phosphate group. So we can see that this strand runs in the 3' to 5' direction. So, and this section, these two strands, they are called the sugar phosphate backbones. Sugar phosphate backbones. This is because they consist of the pentose sugars and the phosphate groups. Also, we say that the two DNA strands are complementary to each other. This means that wherever adenine occurs in one strand, thymine is found in the other. And wherever guanine occurs in one strand, cytosine is found in the other. So the two strands are complementary to each other. Now, the two DNA strands are held together by hydrogen bonding between complementary base pairs and by base stacking interactions. I will explain these two factors. As you can see here, you can see the hydrogen bonding between complementary base pairs. Cytosine and guanine are complementary base pairs. Adenine and thymine are complementary base pairs as well. Now, let's ask a question. Why does adenine pair with thymine and cytosine with guanine? Why not maybe adenine pairing with cytosine or guanine, or maybe cytosine pairing with uh, thymine? This is because adenine forms only two hydrogen bonds and so pairs with thymine, which forms only two hydrogen bonds as well, so they fit. Now, similarly, guanine forms three hydrogen bonds and so pairs with cytosine, which forms only three hydrogen bonds as well. So you can see that they fit each other. Now, any other pairing other than the AT and the GC pairing will destabilize the double helical structure of the DNA molecule. It's just like when two things don't fit, there will definitely be irregularities. Now let's talk about the base stacking interactions. Now, base stacking interactions occurs between adjacent or neighboring complementary base pairs. So these are the complementary base pairs. This complementary base pair and this complementary base pairs are base pair are neighbors. This and this are neighbors, just like that. This and this are neighbors. This and this are neighbors, just like that. Now, in primary schools and even in universities, when we submit our assignment books, we submit it in stacks with one book placed on top of the other. So you can see such arrangement here, a stacking arrangement where you have a stacked base placed on top of the other. But this one now occurs at a distance, and at this distance there is attraction between the stacked bases. So base stacking interactions are non-covalent interactions and include hydrophobic interactions and van der Waals attraction between the stacked bases. As you can see, there is no physical linkage like we see in covalent bonds, but there is definitely attraction between the stacked bases that helps to keep the DNA double helix in order. So the base stacking interactions and the hydrogen bonding between complementary base pairs are responsible for the formation of a highly stable DNA double helix. So, isn't it wonderful and of great marvel how just four letters AT and CG encode the necessary information that is required to build and maintain an organism, including you and me. Just like the 26 letters of the English alphabet are required for the construction of all English words. There is a few more to know about DNA structure. Check the next video.